Thanks everyone for tuning into our presentation. So we're going to be talking about high content screening and the tools we use at Novartis Institutes for BioMedical Research. Co-presenting along with me is my colleague, Yangsha Liu. And we also have Drew Dresser from Amazon Web Services. So before I dive into the presentation, broadly, we're going to talk about just a brief history of apps at NIBR that have anything to do with high content screening. One of the main points we want to drive here is that is why we use Amazon. So as the science changes over time, we have to be ready to be able to scale and manage those type of workloads. And that's what we're going to be sort of talking about, the evolution of the apps over time. Drew's going to take us through the service overview, some of the services that we used in this sort of application ecosystem. We'll dive a little bit deeper into one of the more recent applications that we built called HCS Tiles. We'll talk about the solution architecture, and then we'll wrap everything up talking about the scientific impact. So a short history. People have been running imaging assays and imaging experiments for several years at NIBR. There has been sort of an evolution of tools over time. So this shows a very rough timeline, but if you start from left to right, when we started out, we could visualize HCS images in acquisition format. So basically the format that they were acquired in and nothing else. And essentially being able to look at images that were acquired inside of wells and plates was sufficient, was actually a big win. This was combining all these images from different confocal microscopes. As we progressed, quantification became more important, and the need to quantify at high scale using like an HPC cluster was important. So around 2014, we built an app to analyze HCS images on-prem. But then, as science does, it changed. So cell painting assay technique was published in 2016, and this increased the compute and infrastructure requirements by several orders of magnitude. The storage that we needed, the throughput, the number of compute nodes, the time that we were computing to do the image analysis, everything increased. And that pushed us to seriously look at using Amazon services, which we ended up doing in 2018, running basically high content screening image analysis at scale in Amazon using AWS Batch and other services like Step Functions for orchestration and S3 for storage. And then finally, to sort of wind this all back, at the beginning I said we visualized images in acquisition format. Over time, that became a pain point for people running the assays because they were completely absent the context which is useful, which is an assay context where you're relating things like sample metadata, other type of imaging metadata. So we built another app also native to Amazon to enable users to view these scientific images in a scientific context. So we'll talk about that. So one of the challenges we've had with this type of data is that it just continues to grow, both in pure storage terms, but also in the variety of data. So, you know, Novartis Institutes for Biomedical Research has multiple sites. We have confocal imagers. We're constantly sort of purchasing and upgrading imagers all the time. And as we've gone through this these last few years, the types of data and basically the capabilities have changed. So a lot more volumetric data, Z-stacks, time series, so kinetic imaging, just means a lot of data. And that's put also some stress on traditional infrastructure on-prem. And I'll talk a little bit about how we address some of that with Amazon. So the cell painting assay, which I alluded to. So August 2016, cell painting assay comes out. Shortly after, there was interest within NIBR to start running these types of assays at high scale. So to give you a little bit of background on the scale, so before cell painting, you would typically extract features from an image. They would be somewhat high level. Maybe they'd be in the hundreds at the most. This would be written out into a tabular CSV format, maybe a couple gigabytes, nothing that was too crazy to handle from an infrastructure perspective. What we saw with cell painting is now we were extracting individual features from individual cells within the image. So you could have maybe thousands of cells in a single image, and maybe millions of cells, millions of images in an assay. So what this means in tabular format is hundreds of gigabytes of data, hundreds of gigs of data. You could think of the cell as the row in a CSV, and maybe 2,000 columns, which are showing the features of that cell, like the width of the nuclei. So this puts significant strain on our on-premise infrastructure. So it required a lot of nodes to compute. We had tasks that were running for days. And basically that caused the cluster to be so busy that it blocked other people with other scientific workflows. And then sometimes the jobs didn't complete at all and there was no image analysis results. So it became necessary for us to look elsewhere. So that's cell painting a little bit. If I back up a little bit, we talk about showing images, right? So one of the challenges we had in the beginning was that we had all these different types of microscopes in different sites, and there was no way to view or share the images with other scientists. There was no visualization web service, and the scientific metadata was encoded on image file paths. So the solution at that time was to build a web application with services that integrated the visualization of the images across the different confocal microscopes, provided a search capability for the plates that they were required in and some of the metadata. But there was some drawbacks. One of them was that to view the images, you had to view them in the way that they were required. So you could think of like images inside of a well in a field across channels. So there was no other way to view them. And we hosted this on-prem. Here's what the app sort of looked like back then. It still looks like today. It's still in existence. But essentially, you have on the far left a way to search for either barcodes or folder paths, which the scientists would know where their images were, their plates were. Gives you a list. You click on a link, it would open up a plate in well view, and you could visualize your images. Right. So we move into 2014, and then quantification becomes more important. So the challenges at this point were, well, they had the app to view the images. They had a whole bunch of custom scripts that people were maintaining to manually submit image analysis jobs to extract features on the on-prem HPC cluster. So it required writing a lot of custom scripts, and the inputs to these files were often curated manually or with custom scripts again. So you can think of the input for the cell profiler workflow as a large CSV file. It's just a list of images. So it was error prone, tricky to debug. And finally, there was no way to conveniently share the image analysis results. You had people moving kind of large files around manually. It's not ideal. So the solution was at that time to build a user interface to abstract the submission of the array jobs to the HPC cluster and make it so that you did not have to be an HPC expert. You could focus more on the science and less on the infrastructure. So that was quite a success at that time. And it was built around the cell profiler workflow. But then we had cell painting happen, and this forced us to reevaluate how we could handle that kind of scale. So to handle that, I had mentioned that the cell painting assay was placing a heavy demand on the shared cluster environment. We also had pain points like no centralized logging, job tracking management. So the solution was to re-architect the solution basically using AWS Batch as the compute engine and orchestrate our workflow with AWS Step Functions. We built it in a way where we could have it isolated, but also make it easy to integrate with applications downstream via shared S3 buckets. So we had an app that takes the image analysis results and processes them. So we have a shared S3 bucket, that kind of thing. And then I know it sounds basic, but it was actually a big deal. Centralized logging per cell profiler job in CloudWatch enabled, basically it streamed the output of the standard out from the Docker container running cell profiler for every job running on these nodes. And the reason that's significant is that users submit jobs using this cell profiler protocol, which is basically a list of methods. And it's highly customizable, it's very flexible, but it can also be challenging to debug. You really need access to logs. So we were able to, just by using CloudWatch, give users visibility into specifically what was going on, even on a per container basis for a single image set, which turned out to be of high value because it enabled users to fix problems themselves without escalating it to support. So that app basically displays a list of what we're calling runs, and a run can be defined as an array of jobs that run on a cluster. You can click on any of those links and it will take you to the details of that run, and it will show you the inputs as well as some of the metadata. You can drill down further into looking at individual results that the array jobs ran in. So typically they're submitted via wells. So one well runs on one Docker container. And then, as I mentioned before, we expose the cluster job log, so you can drill down and see like, my method identify primary objects took 9 seconds. That's to be expected. Or, oh, there was a problem. I know what's wrong. Here's how I can fix it myself. Right, so then we finally get to around present day. And again, we had this pain point visualizing the images in scientific context. And this required, building off of the work we did with running the image analysis in the cloud, but also required us to sort of rethink how we managed our workflow and how we basically did our ETL process to prepare the images to be viewed for humans and merge them with other data sources. And we call that HCS tiles. So HCS tiles, and we're going to talk about this more later in the presentation, but what this tool basically does, is that it enables a scientist to view images in a scientific context and basically view them in a way that is intuitive for somebody looking to answer a scientific question. So the challenges were that if you restrict people to view these in the way that they were required, it can provide limited scientific value, or it becomes a hurdle for people to, like, you can imagine people taking screenshots and then pasting it into PowerPoint to share. There's a lot of manual stuff, and it's a lot of opportunity for automation here. There was also no tool at that time to annotate the images and share them conveniently. So the tool addressed that. And then another thing that we'll talk about with the solutions architecture is that these images are typically 8 or 16-bit depth TIFF images. By default, they're very, very black. And what we decided to do was to pre-process them to make them more friendly to the human eye. So along with our scientific collaborators, we developed an algorithm to basically level the pixel intensities for optimal viewing. Of course, that's subjective, but it turned out to be, and Yangsha will talk about this a little bit, that was complementary to having the image analysis quantification, is making something that humans could really see and say, that's my answer. So we'll talk about that more. So I'm going to pass it off to Drew now, who's going to talk about some of the services that we used and how we solve some of these problems. Drew?
Thank you, Nick. As Nick said, I'm going to quickly introduce 3 of the key services that Nick and team use to build their HCS applications. The first that you're seeing on the screen is AWS Step Functions. So Step Functions lets you coordinate multiple AWS services into serverless workflows so that you can build and update apps quickly. Step Functions lets customers like NIBR build data processing and orchestration apps. And they can do this because Step Functions lets you update apps quickly, improve the resiliency of the apps, and actually provide orchestration as a service. Step Functions manages state, checkpoints, and restarts for you to make sure that your application logic is executed in the order that you expect. And on the left-hand side, you can see actually what a visual representation would look like if you were to build your application with Step Functions. This helps immensely with things like development and debugging, because you can get a visual representation of your application and quickly figure out what went wrong or which path was actually executed. In addition, NIBR benefited from features like try-catch and retry and exception handling to automatically perform cleanup or retries within their data processing applications. On the right-hand side, you'll see the serverless polling architecture that NIBR used. So this is when NIBR would use Step Functions to submit a job and then actually poll the status of that job to wait for it to complete. And then once it's complete, it can continue on with the application flow, whether it's a success or a failure. Next slide, please. The next service that NIBR benefited from was Amazon Elastic Search. And Amazon Elastic Search is a fully managed service that makes it easy for customers to deploy, secure, and run Elasticsearch cost effectively at scale. The service provides an open source or the open source Elasticsearch APIs, such as managed Kibana and Logstash. Additionally, similar to Step Functions, you only pay for what you use. So that means if you have the need to spin up an Elasticsearch cluster for development or testing, you can do so and then tear it down once you're done with it, and you're only paying for that time when it's in use. Some of the use cases that we see customers use Elasticsearch for are application monitoring, security, information and event management, and search. And NIBR was able to benefit from this search use case within their architectures that you'll see in a little bit. And if we can jump to the next slide, please, Nick. The last 2 services that I want to introduce are Amazon SQS and AWS Lambda. So going one at a time, Amazon SQS, or Simple Queue service, is our fully managed messaging service that enables customers to decouple and scale microservices, distributed systems, and serverless applications. Traditionally, customers had to manage and operate middleware, but with SQS, you can easily deploy a fully managed queuing service. SQS scales elastically, and again, you only pay for what you use. It has that true serverless model with it. For some fun trivia, Amazon SQS was actually our first service introduced by AWS back in 2004, some 16 years ago now. The second service you see on this slide is Amazon Lambda, AWS Lambda. And AWS Lambda lets you run code without provisioning or managing servers. It's our serverless compute offering. And again, you only pay for compute time that you consume. And similar to SQS, this avoids the need for NIBR and other customers to manage servers. Lambda offers continuous scaling, sub-second metering, and consistent performance for your apps. This pattern that you're seeing here is actually a pattern that NIBR used within their HCS applications, and it shows the integration between SQS and Lambda. And so what customers do with this architecture is you put a message into an SQS queue, and that's step number 1 there on the screen. Lambda service actually pulls that queue for you and invokes your Lambda function when a message is ready to have some work done to it. Your Lambda function would then do the work on whatever it needs to do. And then once it's complete, it returns success to the Lambda service. And the Lambda service will remove the message from the queue for you, kind of completing this end-to-end cycle here, but really allowing customers to scale and control concurrency for their decoupled serverless applications. And with that, I will hand it back to Nick to see how the NIBR team used these services in more detail.
Thanks, Drew. So now we're going to dive into the HCS tile solutions architecture more specifically. And as you can see, sort of the centerpiece of the architecture is the state machine running in AWS Step Functions. It sort of orchestrates everything else like an octopus. But if we start from left to right, a user would connect over Direct Connect and access a web app hosted on EC2 that had some web services also hosted in an auto scaling group. And essentially it would trigger a workflow that would access the images, extract metadata from them, pre-process and create new derived images from them, and then basically report the state back to the end user. So a couple other things to note here. We built another app before this for the image analysis. So we're already storing images in S3. So one of the things for application integration is we have shared buckets where the Tiles app will actually access the images in the HCSCIA VPC, pull them in and process those. If we detect that the images are not available, we'll actually pull them over HTTP from on-prem. So I want to talk a little bit about why we selected some of the services before I jump into the state machine. So if we go from left to right, ElasticSearch was selected, I would say mainly because we wanted the Kibana integration. It also supported all the open source ElasticSearch APIs. It had integration with Logstash. I would say we liked it because the flexible metadata format and the performance. Step functions, of course, is sort of the centerpiece for using this to orchestrate all the workflows going on underneath the hood. It was useful to use step functions in this case because we had a team that did not have a lot of experience using Amazon at first. And state machines was a very straightforward thing to learn because they're defined using JSON. So it's very accessible and you can view you have a visual workflow. So it's very intuitive service and it has a lot of integration with other Amazon services. It's very flexible. Drew mentioned our use of SQS and Lambda. It sort of makes sense to talk about both of them. We made heavy use of the SQS Lambda integration to basically trigger scale out workflows and then control the concurrency from all kinds of things like extracting metadata to actually processing the images. So we used SQS to persist those messages. The trigger integration was very high performance. And you could think of Lambda for our architecture as glues a lot of things together. Also provided a lot of options to tune. And I'd like to mention that it also enabled us to fail quickly, which I think is important to find what works and what doesn't. It just was a joy to use. And then finally, S3, scalable, flexible. It supported event notifications, so you could build ETL off of that if you wanted to, and lifecycle policies to help us manage the data and cost. Okay, so we talked about the high-level architecture, sort of from an airplane. Let's dive into the state machine. So here you'll see the sort of the main HCS tile state machine. We're calling this the create experiment state machine. So what does it do? So when the UI is invoked, essentially the first step is to get a list of images that we need to operate on. So it's making a call, basically invoking a Lambda that's calling some web services, and then it's persisting that list of images in an S3 bucket, right? So after we create the image list, we're actually going to poll, and we're going to make sure that if we don't have the files in S3 already, we're going to transfer them from on-prem. So the sequence here under the hood is basically get the image list from S3, iterate over it. Those become messages in SQS that have a trigger. They call web services. They keep track of the state of the file transfer, and then we check basically the counts of the images in S3 are what we're expecting. As we move on, we have this step in the workflow where we're actually loading basically metadata that we extract from the images as JSON documents in S3. And these messages start as SQS messages, and they get enriched. So we have a Lambda function running ImageMagick, actually, to retrieve the pixel intensities for every image in the experiment, and basically persist those. And then it persists them both in Elasticsearch as well as in custom in individual JSON files in S3 for the next step. Right. So here we're also polling. We're using that polling pattern that Drew had mentioned to make sure that we have the proper counts of these metadata loaded into Elasticsearch. And once that's complete, we're actually going to start processing the images with the custom algorithm that we developed. So again, the trigger here is SQS messages get triggered to get consumed by a Lambda function that's running ImageMagick that's actually going to apply a pixel leveling operation on each image with an algorithm that we developed, and then it's going to persist those derived images, these new images, as well as JSON in another bucket. And of course, we're going to poll for completeness. And then basically, Drew mentioned we used our use of try catch. So obviously, if the experiment failed, we'll report that back to the end user, and if the experiment is complete, we'll also notify via that state change. And that's basically the workflow for when a scientist creates an experiment in the UI, what happens underneath the hood. So I want to pass it off to Yangsha, who's going to talk about the scientific impact that HCS tiles had. I think it makes sense here to tie everything up, because you could sort of lose the forest for the trees by just looking at the architecture. So Yangsha, please take it from here.
Thanks, Nick and Drew. Yes, the two applications featured here have really enabled many cellular imaging-based screening platforms at Novartis. Here, we'd like to share with you two recent examples to give you a taste of what these impacts look like. So the first example is how these apps have enabled a novel screening platform developed by our scientists at Novartis called FluorPack. You can see on the left hand, the pie chart on the left side is the distribution of the fluorescent chemical probes called FluorPack. You can see it covers a wide array of phenotypes. So the neat thing about this platform is that you don't know what you're going to find going in. This allows our scientists to make unbiased discovery of their processes and functions that of interest. But on the other hand, this poses a major challenge to image quantification, because traditional segmentation-based image quantification relies on this kind of prior knowledge. You have seen that Nick has presented to you the application called HCS IA, developed by the teams at Novartis that allows you to quantify your images at scale. Luckily, this application has already have a segmentation-free algorithm incorporated into it. So that allows the quantification of those FluoroPack images at scale possible. And at the same time, we also build a AWS Tiles application allows for efficient manual inspection of those images, which is very essential for this type of assays because of the need to discover new biology without prior knowledge of what you're trying to look for. So on the right-hand side, you see a typical workflow of a FluoroPack experiment. So you have the wild type. This one actually represent a typical way of using the FluoroPack platform. You have your gene of interest knocked out, so short named KO, and the wild type is short named as WT. So in the KO line, your protein of interest is removed from the cell line, so it does not have that protein. And both of those cells are plated on different wells on the same plate to facilitate comparison. And the probes are plated on the plates, one probe per well. And after incubation for a certain period of time, those cells are imaged by confocal microscopes, and the resulting images are going through quantification, and then some clustering and ranking methods are used to rank these probes. So without a tool like AWS Tiles that allows our scientists to review images efficiently, the scientist will be limited to only reviewing those images of the top hit probes. But with AWS Tiles, we gave our scientists the luxury to review all the images of all the probes. This really helps those two processes to help each other, complement each other. The manual inspection can validate and inform the automated pipeline to allow our analysts to adjust the analysis parameters, making the analysis more accurate over time. So next slide, please. Let's take a look at a concrete example here. This is one of the proof of concept experiments used to establish this FluoroPack platform. It's an outcome of a T-MAM41B screening. On the top of the figure, you can see on the left-hand side is the ranking of all the probes, the 44 probe panel. And on the left-hand side is the zoom-in view of the top 15 probes. The ones that has a star are those validated by manual inspection. The bottom half shows 4 representative probes. On the left-hand side are 2 probes that didn't show any phenotypic changes. The group on the left, the left side of the group on the left are the t-SNE plots of the whole screen with the corresponding probe zoomed in. You can see from the t-SNE plot, there is no separation between the knockout cell lines and wild type cell lines. And same thing with the other probe. And the images confirm that. On the right-hand side are 2 probes that actually show reviewed phenotypic changes. And from the t-SNE plot on the left, you can see the clear separation between knockout cell lines and wild type cell lines. And on the images on the right gives you a clear confirmation of that. You can see in the knockout cell lines, the lipid probes accumulated as punctate, while these punctates are missing in the wild type cell lines. Or just to quickly mention, the blue are the DNA and the greens are the probes. Okay. Next slide, please. Oh, this is another example. That is an RNA screening for genes involved in tau aggregation. So tau aggregation is a hallmark of Alzheimer's disease. Clearly, studies like this can help bring us closer to cure for such a devastating disease affecting millions of people in US alone. So on this, in this experiment, siRNA, small interfering RNA, are added to the cells to specifically targeting genes to knock down those genes. So in this group, you can see on the left-hand side, the bar chart are the quantitative results, and on the right hand are the images as seen in ACS tiles application. The non-targets are actually scrambled RNAs serve as a reference to establish the basal level of tau aggregation. And the MAPT is actually a tau siRNA that knocking down tau. So in here, we have tau protein wiped out as negative control. So here, again, the blue channel is DNA, and the red channel are the tau aggregates. So for this group of gene, you can see from the bar chart on the left, knocking out those genes increases tau aggregates. It's very significant increase. And on the right-hand side, you can see the images really confirm that result, indicating these 4 genes might be involved in suppressing tau aggregation. So next slide, please. This shows another group of gene has this opposite effect. On the left-hand side, again, the bar chart coming from quantitative analysis. And so knocking down those genes with siRNA decreases significantly the tau aggregates. And the images on the right shows confirm the quantitative results. So this really shows how the ACS-IA and ACS tiles complementing each other. With this experiment, so we know exactly what kind of phenotype we're looking for. So the segmentation-based quantification is very effective. With ACS-IA, this process can be highly scalable and also very reliable. And the ACS tiles complement that with efficient image review. These experiments involve like using reagents directly from Alzheimer's disease patients. So being able to see all the images is especially valuable for this experiment. So next slide, please. Yeah, here is the ACS tiles application. As you can see, it displays very high quality images with the experimental context. And you can also, Nick has mentioned that there's a lot of pre-processing of these images. One of the pre-processing is actually bring related images to the same intensity range, so you can have apple-to-apple comparison. And these images can also be directly copied into your presentation or your publication manuscript for scientific communication. We also have live report pages that you can send link to your downstream collaborators directly to collaborating and documenting your observation. So it's been a great application helping our scientists. So next slide, please, Nick. Yeah, so I'd like to summarize this talk by these 2 slides summarizing the impact of ACS tiles application. So I do see like the tools we build have like impacts in 2 different levels. At the basic level, we make the existing activities more efficient. And at the higher level, we actually enable previously impossible activities possible to allowing our scientists to break new grounds in their research. So for the FluorPack assays, ACS tiles turns 8 to 10 hours laborious like chore over 2 weeks into a 2-hour really engaging discovery activity. And in addition, without ACS tiles, the image review will be limited to only those top hits probes. But with ACS tiles, all probe review previously time prohibitive task is highly possible and also very efficient. And next slide, please. We want to kind of briefly give you an overview of the standard screening assay benefits we get for ACS tiles. So it actually turns a 4 to 6 hours task into a 1 to 2 hour task. And it also increased the number of compounds you can review from 20 or so to like hundreds. And this also allows like previous impossibly time-consuming all hits review turn into a very efficient and a very scalable task. So with this, I'd like to say we have showed you how at NIBR Institute for BioMedical Research our scientific innovation pushes new requirements and drives our evolution of our applications, and how our informatics teams leverage software platforms like Amazon Web Services to effectively evolve our application to keep up with the pace of the innovation. And with that, I'd like to thank you for your attention and thank the LC HS team for giving us this opportunity to present here, and also thank the our NIBR colleagues for making all of this possible. It has been really fun being in this process. Thank you.